Good afternoon, everyone. So we make a start. Thank you for joining the QAAFI seminar series today on the 10th of May. Next slide, please. I'm your facilitator today. Welcome. Moving forward. And first, I'd like to acknowledge the country. The University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. And next, just a few housekeeping announcements to make. The seminar goes on till 1 o'clock. We usually have the questions at the end, and please use the Q&A tab for your questions, not the chat. The chat won't be monitored. So please remember to use the Q&A, and remember that we'll go through the questions at the end of the seminar. And so today I'd like to introduce Professor Mary Fletcher. Mary is a natural product organic chemist and leads the natural toxin group within the Centre for Animal Science at QAAFI. She's previously worked as a research chemist at UQ and also DPI Biosecurity Queensland before joining QAAFI in 2010. And Mary is also an affiliate professor in the School of Chemistry and Molecular Bioscience and also a fellow of the Royal Australian Chemical Institute. In 2016, she was elected to president of the Queensland branch of the Royal Australian Chemical Institute. So today, I'll hand over to Mary and hope everybody enjoys her seminar.
Thank you so much, Arla. I'm hoping I'll get the technology going now. So today I'm just talking about some of the work we've done with indospicine, in particular, some work we did last year in relation to a spate of dog poisonings in Victoria. So as well, this was an investigation carried out by the Victorian government where a whole lot of dogs had died, and the original source of the poisoning wasn't clear, but the one that frightened us the most was they actually thought it was beef. So it was a very emotional thing because these are domestic dogs that were dying, people's pets, and anyone who's got a pet can fully understand that this is really an emotional time. People want to know the answers as to why they were dying. And when it became quite clear that there was a large number of dogs that were dying, there was in total, there was at least 68 dogs that were severely affected, and 26 of these dogs actually died from this poisoning. So the investigation was carried out down in Victoria. The cases were down in Gippsland for the eastern part of Victoria. And the Victorian government set out to identify what exactly was responsible. What was the toxin that was causing these deaths, the source of the toxin, and basically where the toxin was coming from. As the title of my talk would have elucidated, indospicine ended up being the toxin involved. So what I'd like to do is take you through some of the research we've done in poisonous plants, particularly relating to indospicine, and then come back to the dog case and explain what we actually did that actually helped the Victorian government solve what was actually happening with these dogs. So for the past 20 odd years, I've been working in a field that we term natural toxins in agriculture. These are toxins from natural sources, basically things like poisonous plants, fungi, cyanobacteria. A large part of my work has been focused on toxins in poisonous plants that affect grazing livestock. So the pretty pictures on the right where the cows and the sheep are in paddocks with pretty flowering plants, those are all toxic plants that the cattle and the sheep are hopefully avoiding, but of course the abundance of them makes it difficult. So cattle, the livestock that graze these things, can end up with livestock losses, poor weight gain, death and reproductive losses. But the other insidious aspect of our research is the toxins that can form residues in meat and other products. Other products can be things such as milk, which is clearly important for trade. The other natural toxins we've worked with is mycotoxins, which can affect health, either human or animal, and also trade. So these are fungal toxins that grow on grains. They're often regulated by legal limits. One aspect of the natural toxins is they're typically not regulated. These things occur sporadically. They don't generally leave residues. So my background, as Ayla said, is in chemistry. So we're looking at chemical analysis of these toxins. So my slides have a few chemical structures. So some of the things that we looked at are protein alkaloids, the blue heliotrope on the left there, or the fireweed on the right. So that's what's commonly called fireweed. They're a Sneakio species. Bracken toxins, that are quilicides, and the one down at the bottom is the indospicine, which is common in the Indigofera species. And we'll talk a fair bit more about that during this talk. So as I've said, there's a whole lot of diverse chemical structures. Some of them are quite complicated, but we're trying to analyze these both in the plant samples and in the animal tissues. So our sheep here grazing in the paddock with Patterson's curse can actually accumulate some of the toxins in their tissues, and we've developed methods to look for these toxins. Typically the structures over here, which are the protein alkaloids. We've also done more work recently looking at the same toxins in honey, in that the bees, which are just another form of livestock in essence, actually visit the same plants and transfer toxins into the honey. So my interest with indospicine stems back from when I first went back to DAF in 2003. So it's quite a while ago now. So we had a large project that looked at the natural toxin residue risk. So we were looking at 92 different types or groups of toxins to which cattle are exposed across 11 different regions across northern Australia. We looked at plants, fungi, cyanobacteria. That eventually was published in a large report, which in many ways resembled a thesis back in 2008. So the at-risk assessment, we looked at the residue risk of the toxins and the plant sources, and we assessed these across the 11 regions. We looked at the distribution and abundance of the plants. Things that are more widespread than others were obviously of greater interest. Whether the plant was palatable and readily consumed by the livestock, the actual livestock exposure, was it in the pasture that the livestock actually grazed? The toxin itself, the concentration, and the residue persistence. A lot of toxins an animal might eat, ingest, and it will be excreted from the system. So we were looking at a trade aspect, so the residue persistence was of great interest. And the market type, whether the animals went for live export versus slaughter. An animal that goes for slaughter basically captures the toxins that are in the body at that point in time. If you send an animal on live export, the toxin can be excreted from the system during the time of transport. And after all of that work, what we did conclude was that in contrast to most other toxins, there is a likelihood of indospicine residues in livestock across all regions. That was a major assessment. It took a lot of work to get there. We did feeding trials with cattle and all sorts of other aspects. But all 6 of the above criteria, indospicine was seen as a risk, because it is readily, the Indigofera plant is readily consumed by livestock. It's widely distributed, and it performs very persistent residues in the meat, which we'll discuss as we go through. So what is indospicine? It's a toxic non-protein amino acid. It's an analog of arginine. For anyone who really likes chemical structures, arginine's a simple amino acid, one of the essential ones. It has an NH group at this internal position. Indospicine is exactly the same molecule, except it has a carbon at that position. So a very similar structure. which is partly why we believe it is so toxic, in that the enzymes accept indospicine as if it was arginine, but they can't process it. So as the text says, it's a non-protein amino acid, which means it's present in both plant material and in animal tissues as a free amino acid. And it's also unusual that it accumulates in animal tissues and forms these persistent residues. It only occurs in Indigofera plant species. There are more than 700 of these worldwide, predominantly in tropical and subtropical regions. And the highest level of indospicine is found in these two, Indigofera spicata and Indigofera linnaei, which are commonly known as creeping indigo and Bairnsdale indigo. This is one of my holiday snaps where I actually found both of these plants growing side by side on a footpath. So the plant on the left may be familiar to people who are keen gardeners. It occurs widely in lawns in southeast Queensland. It has some very characteristic seed pods and pink flowers. It has a large taproot, which any good gardener will struggle to get out of the lawn, but it is widely present in our lawns. Fortunately, it hasn't as yet got into so much into pasture. It's actually an introduced species that was introduced into Australia when researchers were looking for fodder crops. It's highly palatable. Unfortunately, it has this toxicity aspect, which is why they discouraged releasing it. The plant on the right, however, is Indigofera linnaei. It's a native species. It's prevalent in pastures all across northern Australia. So there's a couple of distribution maps there. The bottom one's the herbarium map, which shows where people have collected plant samples across northern Australia. So it occurs remarkably from the arid desert regions of central Australia to tropical regions with much higher rainfall. But in these arid regions of central Australia, it tends to be more a problem because it can at times be a predominant pasture plant. It has a long taproot similar to the spicata species, and it's one of the first plants to respond after rain. And as a consequence, it has seasonal high prevalence in pastures in central Australia, particularly when other pasture dies out and you get the early summer rains, and this is the first plant that regrows. So after we'd done our risk assessment, it probably wasn't a surprise when we found an actual case where people had reported dogs that were dying from eating camel meat. And we were asked to investigate because the histology looked like indospicine. Also, these animals had been consuming a known diet. The dogs in question were pedigree dogs that are being treated for a skin allergy. So they were on a prescribed commercial diet of camel meat, which was mixed with sweet potato. And they'd been fed this exclusively for between 30 and 139 days. So all the dogs that presented with the severe hepatic disease, there was only 4 initially, a larger number developed over time. But they all had elevated liver enzymes. They had jaundice and had characteristic liver histopathology. Now, I'm not a pathologist, so I claim no interpretation of the pathology, but my colleagues in that investigation were. So there was severe and extensive hepatic damage with only small areas of viable hepatocytes, and the majority of the hepatocytes have been lost and replaced with areas of hemorrhage. What was very good about this investigation was that this histo was actually recorded in the papers, but it was also published online, so that in later investigations, the pathologists were able to draw on these reports and actually look at the slides that were being produced. So in the dogs that had died from eating the camel meat, we received samples of muscle, liver, plasma, and some of the diet. So indospicine was present in all of the dog tissues. So we were pretty clear that it was indospicine. There was a small amount of it in the camel meat. Unfortunately, because these dogs had been fed this diet for some time, there wasn't actually historical diet batches. So it's possible that some of the earlier camel meat had actually been higher in indospicine. Our initial investigations measuring indospicine, we just had an LCMS method where we were looking for indospicine itself, but we found that because of the complex nature of the matrices, sometimes the mass spec signal was increased or decreased due to these matrix effects. And as a consequence, we now use an isotopically labeled indospicine as an internal standard. So we've got a deuterated indospicine that co-elutes with indospicine. So if the signal's enhanced or decreased, the deuterated one is similarly affected. So as a result of our camel meat studies, we detected the indospicine in the camel meat, and we concluded that it was more than likely Indigofera linnaei, which remarkably has been reported to be a preferred camel food. So there's a large number of camels that range around Central Australia. On the map up the top there, the lighter colour is the area in which feral camels are found in Australia. And the green star indicates that where the camels that were actually fed to the animals had come from. So as I said before, indospicine only found in Indigofera species. There are about 60 in Australia. So just to confirm whether or not there are other ones out there, we have done surveys across more than 15 species. The only ones that we actually found levels of are the 4 species listed there. And again, the ones that are clearly the most problematic are the Indigofera linnaei and Indigofera spicata. Spicata, as I said, is the one that's in our lawns in southeast Queensland. The dots on that map represent the current spread of that plant. It was introduced and released in the southeast Queensland region. It is increasing in prevalence, but as yet it has never been into pastures in the more arid regions. And at this stage, it doesn't represent the same risk as linnaei, which is obviously more widespread. So the levels of indospicine linnaei, we also went and had a look at what the seasonal effects of that were. So it's highest in the new spring growth following earlier rain. So that's just a plot of samples that were collected at a number of different sites in Central Australia over a period of months. There was early rain around September, October, and then a more intense rain at the end of February, towards February. But you can see that the levels of indospicine were much higher in all the samples in the earlier collected samples. Now, for those of you who aren't aware what the pasture looks like in Central Australia, these are wonderful pasture photos by one of my collaborators. That the top series of photos are taken at exactly the same spot as the second set of photos, which were after that rain, as we mentioned in February. So in November, you've got pasture, if you can call it that, and most of the plants are these Indigofera linnaei. So the risk to any animal, if there's not much pasture available and the linnaei is available, that's what they're going to eat. It's highly palatable. Whereas in the later part of the season where you've got much more pasture growing, then even though the linnaei is ending up being larger, it's actually a proportionally a lower amount of the pasture. So Eddie Tan is one of my students who actually did one of these studies with feeding Indigofera to camels, just to verify what we thought was going on with the persistence of the residues in the meat. So these are our beloved camels that we used in the study. There were 6 relatively young camels. We fed them a known dose of Indigofera spicata because that's what we had available. And we knew how much indospicine they were being fed. And we fed them on that diet for 33 days. During that time and throughout the trial, we collected muscle biopsy samples and blood samples at 7-day intervals. Surprisingly, collecting a muscle biopsy sample from a camel was not too traumatic, but our camels certainly had a needle phobia, and collecting blood samples is something I don't really ever want to repeat again with camels. So 3 of these camels were euthanized at 33 days, and the remaining 3 were fed an Indigofera-free diet for a further 100 days. So we analysed the indospicine levels in the plasma, the muscle biopsies, and the necropsy samples. So I've mentioned before about indospicine being persistent residues. So these are the indospicine levels. In the green is the plasma, and the red dots are the muscle biopsy samples. So you can see the indospicine goes up during the feeding phase. It certainly reaches a peak, and it goes no further after we stop feeding them, but it did not deplete very readily. So at the 100-day mark, there was still detectable indospicine in their tissues. So from this data, you're able to obtain the half-life of depletion, which is it's more than between 2 and 3 weeks for both the levels in the muscle and the plasma. So this means that you can actually have indospicine residues in tissues of an animal that hasn't eaten indospicine in the last 3 months. The levels we fed this in this trial were not particularly high, and it's really expected that the levels could be much higher in the wild. There have been other animal species trials done. As part of our original natural toxins project, we carried out cattle trials. There were horse trials carried out in Alice Springs, where they again fed Indigofera linnaei to them, and rabbits and goats at the University of Queensland. So all demonstrated the similar accumulation of persistent residues of indospicine in muscle and serum. And our conclusion was that it was more than likely that all animal species consuming these Indigofera species would accumulate indospicine in their tissues, and then indospicine residues would persist long after consumption has ceased. So this takes us back to the dog cases in question, which was an investigation in Victoria. So as I said before, we had actually published the histopathology from our camel cases where their dogs were poisoned. And it was this published histopathology which actually enabled them to identify the toxin responsible, well, to suggestively identify the toxin that was responsible for the cases in Victoria. So they looked at our slides that were published, and they were quite keen to believe that the histopathology they were seeing was similar to what had been reported for indospicine. So I received the phone call where they were requesting our assistance with indospicine analysis of both dog tissues and pet food samples. So starting from their presumptive diagnosis of indospicine, these samples arrived up here in our QAAFI Natural Toxins lab at Coopers Plains, and LCMS analysis confirmed the presence of indospicine in the euthanized dogs. So the levels in the canine sera and the liver were similar to what we'd seen in the dogs that were poisoned by camel meat. So it seemed pretty well tied up that indospicine was the responsible toxin. However, the pet food samples that were sent in the initial investigation all tested negative for indospicine. Unlike in the camel case, we actually didn't know which of the dog foods that were being fed was actually the responsible diet. Remembering that in the camel case, the animals had all been fed the same diet at the time, which is not the case when you've got 68 different dogs being fed by different dog owners. So as soon as we had these results, we had the media was onto this as well. So it's an interesting experience to be having an investigation where no sooner had we reported the results back to the Victorian investigators, but it was appearing in the press the day after. As I also mentioned previously, we'd actually looked at a lot of the Indigofera species around Australia. There is one that's quite common in Victoria called Indigofera australis, but our analysis has told us there is no indospicine in this species. So our Victorian colleagues initially thought that this might be the cause of their problem, but we were pretty clear that there was no indospicine in that plant. So the question then comes, if there's indospicine in the pet food, where was it coming from? And which batches of pet food were responsible? The work we've done previously on the fact that indospicine is cumulative and persistent means that the dogs also could have been consuming the toxic meat days, if not weeks earlier. So what we suggested was that the Victorian investigators investigate what batches of pet food were available that were from a previous period. And so in this case, they were able to find dog owners who had kept food samples from batches that they had bought earlier. So we then got a number of samples that did test positive for indospicine, and we had ones up to 13 megs per kilo in some older samples of pet meat. So again, we've now getting lots of reports about the dog meat being recalled, and there was a focus on Northern Territory horse meat. Initially, everything we had samples was either called beef or kangaroo meat, and it raised some concerns as to what the pet food really was that was causing the problem, because we had so many different dog owners, and it wasn't clear what the common factor was. So much of the investigation on the ground was always carried out by the Victorian state government. So they eventually traced back to a shipment of horses that had arrived from Central Australia. Some irony in this was that the horses were actually intended for the abattoir in Queensland, but were diverted to Victoria by the COVID state borders. So COVID plays a role in just about anything around this country. So horses in Central Australia, there are vast numbers of feral horses in the same way there are feral camels. The feral horses, current estimates are around the 500,000 mark. My colleagues in Central Australia claim that they could be much more, just from the sheer numbers that they see. Horses, like camels, are not as sensitive to the hepatic effects of indospicine, but like camels, they also accumulate indospicine in their tissues, and this is the problem in this situation. Indigofera plants actually contain a second toxin, which is called 3-nitropropionic acid, or 3-NPA. So it's also found in Indigofera linnaei. It's also found in Indigofera spicata. And as a result of this neurotoxin, the horses actually suffer from a neurological condition called Bairnsdale horse disease. So one way that it's been dealt with in the Northern Territory, because they know that the fact that the horses accumulate indospicine, is that they use Bairnsdale horse disease as a sign of indospicine exposure. It's not caused by the same toxin, but the toxins are co-occurring. So we actually received blood sera from horses on the property, the suspect property in the Central Australia, and showed serum levels up to 42 megs per mil of indospicine. So they certainly had exposure to Indigofera and had been consuming it. The veterinary inspection confirmed that the horses were not suffering any apparent signs of the neurological condition that called Bairnsdale horse disease. This is a condition where they have general weakness, nervousness, incoordination, twitching, swaying, and characteristic toe dragging. So the horses, as they appeared normal, were actually shipped out of Central Australia because they didn't believe they were suffering from the horse disease and therefore shouldn't have had indospicine in them. Again, full press coverage of the investigation. So at this point, we'd confirmed the toxin was indospicine. We'd found the indospicine-contaminated pet meat, and the Victorian colleagues had traced it back, and we'd confirmed that the horses from Central Australia on the property that they believed was the source property did actually have indospicine. At this point, there was considerable concern raised about the labelling of pet food. So the indospicine contamination in Victoria has been confirmed by this investigation to come from horse meat from Central Australia. However, it's worth pointing out that none of the samples that we tested at QAAFI were actually labelled horse meat. And the pet owners were particularly concerned because most of them said they wouldn't have bought horse meat. They thought that they were buying beef or kangaroo pure meat. So one of the things that many people are not aware of is some of the under the Australian standard, the manufacturing and marketing of pet food. There is an alarming section 3.13, which reads, if a meat ingredient constitutes 25% or more of the meat component and is the main meal ingredient, then this meat may be referred to as the variety name of the product. And the example that's given in the standard is where beef is the main meat ingredient in a product and is present at more than 25%, then the variety can be named beef. To me, this is concerning in that there's clearly a difference from a mince sample that's labelled beef as one that's labelled 100% beef. That investigation, the concerns around that, I believe, are still ongoing. So that concludes where we were up to with our investigation. Our side of it was purely the analysis of indospicine in the samples. So any further investigation of this has been carried out by the Victorian government. And so my final slide is about suggestions for minimising the risk. As is shown by the dog on the side, our domestic dogs don't find their own food. The pet owners provide the food. So in the camel meat scenario, the dogs had eaten the same meat because they were under a treatment regime for up to 4 months. And in fact, in all cases where indospicine toxicity is reported, affected dogs have been repeatedly fed the same diet over a prolonged period. It's also worth noting that dogs are particularly sensitive to indospicine. And since meat represents a large part of the canine diet, there is an increased risk from indospicine-contaminated pet meat. So in all things, providing a varied diet is one way of reducing the potential risk. As humans, we don't eat the same meat day after day after day. And nor if we eat a piece of steak, it won't be the same piece of steak or the same piece of whatever other meat as what we consumed last week. It'll be from a different source. So what a number of pet owners do is buy food in bulk and then feed that same feed over extended periods. And this actually increases the risk if that pet food was to be contaminated. I'm not saying that a lot of the pet food is, but it's just a similarity between all the cases that we've seen. I'm often asked about the risk to humans. If you look at the major risks, dogs are particularly sensitive to indospicine. We've done studies with hepatocyte cells, and the dog hepatocytes impacts are far greater than all the other species we've looked at. And we have actually looked at human hepatocytes. It's difficult to do a human feeding trial, but the closest we could do was doing a cell study. So human hepatocytes are not as sensitive to indospicine as canine ones are. And of course, as I mentioned before, the meat is not as large a part of our diet as a dog, where it's the primary component. So it remains for me after this to acknowledge the contribution of both Gabby Netzel and James Peacock in this study. There's a lot of other people who obviously have contributed to our indospicine studies over the years, but Gabby and James were the ones who were in the lab doing the analysis for the samples that were coming from Victoria. Because of the urgency, there was often the request that these were dealt with on the same day or the next day. It was full on for quite a number of weeks trying to get these samples analysed. So Ala,
yes, thanks, Mary. That was really interesting. There's a question in the chat, and I think you've more or less answered it, but you can, confirm that do herbivores, camels and horses become ill from eating the Indigofera plants, or is it just the carnivores that eat the meat?
Okay, the camels and the horses can suffer the liver toxin effects. They're just not as sensitive to it. We have done some trials where I think there were some local horses where Selena, Aussie driver, I'm not sure if Selena's on there, but Selena did the histo on those. The horses actually became quite ill eating the Indigofera, and they had levels in their tissues that would have killed a dog. So we looked at the histology of the horses. So they did have effects, but it wouldn't have been enough to kill them in the same way it kills dogs. And likewise with the camels, with the feeding trial we did, we looked at the livers. The camels that we euthanized were quite healthy. We were just wanting to look at the different levels in the different tissues. So there was indospicine right through all of their tissues. It was in the liver. We could see some signs of histo, but certainly not enough to cause any ill effects.
And there's a question about the results of the study. This is from Pat now. The first one was from Barbara Williams. Have the results of this study raised concern about Australia's international trade of meat or live animals? Are overseas markets concerned with this issue?
That's an interesting question, Pat, because that was actually the same reason why we actually did the original natural toxins project back at ARI back in 2003 to 2008. There was a concern about how international markets would view our meat, largely because our plants, we have different toxic plants for everyone else in the world. So some of these plants were novel, and the fact that you can ban international place restrictions on markets just based on, Pat and I probably both lived through the trials of when there were contamination of pesticides in meat, and there was bans from international markets. So yes, there's concerns about it. And that was why I was largely concerned when they said it was beef. The same regulations, obviously, about pet food and human food aren't the same. So the cross-contamination with different species of meat in your mince is not a concern. So there was coverage internationally. As soon as these cases, there was certainly international press coverage of all of these cases of the dogs dying.
There's also a question from Ali about, he's wondering if the wild camels and the horses don't show severe signs, is it possible that they have a natural adaptive response? Being prey animals in the wild, I'm not quite sure what he means in that part. So do other,
yeah, they don't show the same signs. The horses definitely have the neurological signs from the 3-nitroxypropanoic acid, but they don't show the same liver toxicity. I don't know what the evolutionary aspect of it is. Dogs are known to be sensitive to many toxins that humans aren't. a classic example is chocolate. You should never feed your dog chocolate because they react to it. Whereas for a human consumer, I think I can eat a hell of a lot more chocolate than they can. It's worth noting that the wild camels and horses aren't native to the they're all introduced species. So there's large populations.
Does it ever end up in kangaroo meat?
In kangaroo meat, we've never done that investigation. From the number of species that we've fed Indigofera to and seen the accumulation, if they eat Indigofera, I would assume that they accumulate it.
I was just thinking more from the point of view from something that was indigenous to Australia, whether it is just introduced mammals or, anyway. But,
we don't know. We've never done it. When the Victorians said that there was kangaroo meat, I thought, here we go, we're actually going to get to do this one. But no, it didn't turn out that way.
But there's another question here from Harry Lamb. You mentioned the use of Bairnsdale disease, an indicator of possible indospicine contamination in horses. But the horses in 2021 didn't have that disease.
Yep.
So is it likely they'll introduce random sampling of horses for the indospicine, or is that something you already do? Or
No, they don't sample. That has actually caused considerable concern in the Northern Territory government. The Northern Territory Meat Act actually has restrictions on what you can sell, where you can sell horse meat, where you can slaughter horses from, and where you can sell them for either pet meat or human meat. But it's all based on Bairnsdale horse disease. So yes, the fact that it wasn't evident in this case has actually caused concern to the Northern Territory government. And I don't actually know the answer, but they are considering that as we speak.
So it's nothing that's tested.
It's not routinely tested, no. We're probably the only lab in Australia who are capable of testing indospicine at that scale.
So it's not tested by any.
No, none of the residue labs do it, no, which is why we ended up getting the phone call.
Yeah, fair. Well, sounds like it should be something that's tested. There's a comment by Barbara or a question wondering if there's a microbe that could be involved in the breakdown of the compound.
Barbara, we did do a project with DAF. Gabby had an advanced Queensland fellowship to study the microbes in the gut of the cattle that would seem to be breaking down the toxin, breaking down indospicine. We weren't able to actually culture. They certainly did break it down in the rumen system to some extent. We could increase it, but we still weren't able to actually identify the microbe. One of the interesting studies that Eddie Tan did was actually looking at breaking down indospicine with alkali. If you heat it up with bicarb, you could actually break down the indospicine. So it's possible to actually decontaminate it out of pet food entirely. I guess that was one other option that was considered. But of course, it's not widespread in pet food. It just happens to occur in these episodes.
So that pet food law, that 25% minimum for description of a animal protein source, it seems a bit, what's the word, inappropriate. Is that something, do you think, that might change, or?
It's under consideration because there was a fair bit of backlash from the public based on the fact that what they thought they were feeding their dogs wasn't what they actually were feeding their dogs.
Well, but the pet owner and dog owner, I'd be seriously annoyed with that.
I figured you were Alan, giving your lover your little puppy.
She's been barking, by the way. I've been going mute for that reason. She must be concerned about what you talked about.
Yeah, no, it is. Obviously there's also the labeling of the ingredients. Generally people, the labeling will actually state what percentages of what's on there. But
so it should have this percent horse, this percent camel, this percent beef.
That's the ingredient label. There's the other label that's on the front. Yeah. Even the tin pet food, if you look at something that's labeled beef, it could be have beef as the major ingredient.
Yeah.
But if you look on the back of those, you'll actually see the fuller list of ingredients. It's just unfortunate that in some pet food shops, they'll just sell something as beef without the
Yeah, I recommend chicken necks. It's obvious it's not a neck from a turkey or anything else, but some advice there for pet owners just from me.
Well, we certainly don't know where chickens eating Indigofera.
Anyway, there are no more questions online at this point. And just if anyone else would like to ask a question, you can ask now. Otherwise, we will wrap up and advertise next week's seminar. Thanks, Mary. I guess I did kind of wonder about something. I don't know if you know the answer to it. Why does an amino acid that doesn't form a protein exist in the wild plant like that? Like what, you said you didn't know the evolution, right? And so you don't know the evolution, but are there other toxins out there that are in plants that are random amino acids like that? The
amino acid one, there's not. A lot of plants do produce, well, they're believed to produce toxins as a defense against herbivores, particularly right. Like the photos I showed you originally of the colored flowers. They're like warning colors that, you know, we contain things that are bad for you, so that there are certain insects that will eat them, but most insects can't tolerate the toxins in the plant. Unfortunately, right. The livestock are actually similarly just herbivores, so they're eating a plant that's, you know, got a self-defense mechanism. The indospicine, really, I can't answer that because it's not acutely toxic. It's just because it builds up in the system. It doesn't actually, if you were to design a defense mechanism for a plant, it doesn't actually make that much sense.
Yeah, so it just builds up if you eat it all the time.
Yeah, you eat it all the time and it builds up in the tissues.
Kieran has just asked a question about whether the toxin can be used for other things like invertebrate pest management. Would it be something that could make, I guess, ticks and flies sick or something? I don't know how, whether...
I think what Kieran's talking about is the alternate to 1080, the bait that they put out there for dogs and foxes and things.
Yeah.
It was considered that, but the veterinary advice was that the dog death was actually more inhumane than the 1080, so they didn't want to do that. Yes, but because it is so targeted at canines, it does actually have that potential, but it's been considered and rejected, I guess.
Yeah, okay, so yeah, all right. There's also one guest who's asking if your presentation can be sent to attendees afterwards.
I believe that it's recorded, so the recording becomes available.
You'll get the recording. John, you'll get the recording of the presentation afterwards. Is that okay?
Yep, should be a link to it. Sent.
And then, so thanks, Mary. That was really enlightening for me anyway, and happy with how I feed my dog. Good. So next week we've got Professor Mark Turner, and he's going to talk about the applications of lactic acid bacteria to enhance food safety, reduce food spoilage, implement plant-based dairy alternatives. I believe these are with SAFS, School of Agriculture and Food Science, and he's an affiliate with the Centre for Nutrition and Food Sciences and Coffee at UQ. That's all. I'm just checking if there's any other QAAFI communications would like to answer. Do you want to answer, Jackie?
No, that's all. Thank you very much, Mary and Ala. It's great.
Okay. Thanks, everyone. Thanks for joining. I will see you next time at another exciting QAAFI seminar presentation. Bye.
Bye, everyone.
Thanks, everyone.